Both gene bank and botanic garden conservation programs preserve plant shoot tips in liquid nitrogen. These cryopreserved collections serve as backups to vulnerable plants in fields and in the wild. Shoot tips are often more difficult to preserve than storing seeds, but necessary when seeds are unavailable or do not represent the desired genetic composition. Many institutions, including the USDA National Laboratory for Genetic Resources Preservation, rely upon vitrification methods to cryopreserve tiny 1 millimeter shoot tips from many plants, including species of grape and peppermint. Successful plant cryopreservation techniques rely on the removal of water from plant cells and tissues prior to storage in liquid nitrogen. Shoot tips contain a lot of water, which forms damaging ice crystals when it freezes. Therefore, water must be removed prior to liquid nitrogen exposure. This is tricky because shoot tip cells need to be hydrated to stay alive. Adding cryoprotectants allows for water to be removed and then replaced with other solutions that change the freezing behavior within a cell while keeping that cell alive. These protected cells enter a vitrified state when cooled, rather than forming ice crystals. Vitrification is the formation of a glassy state as the result of rapid cooling of a solution. While many parameters contribute to the successful formation of a glassy state, the viscosity of a solution and cooling rates are most important. So what is a glassy state? A cell in the glassy state can be best thought of as a bowl of gelatin. See how it keeps the fruit in place? Much like gelatin, vitrification will keep the organelle suspended within the cytoplasm of a cell. If shaken, these objects can still move slightly or vibrate within the gelatin. The glassy state formed within the cytoplasm of cells prevents ice crystals from forming in freezing conditions, which improves cell survival. Thereby, the formation of this state is crucial to successful cryopreservation. In many vitrification techniques, plant cells and tissues are immersed in highly concentrated mixtures of cryoprotectants before they are plunged into liquid nitrogen. These cryoprotectant mixtures serve two roles in the preservation process. One, to remove water content and increase the viscosity of the cytoplasm of the cells, which is essential to successful vitrification. And two, to protect the integrity of the cells themselves. The two most commonly used cryoprotectant mixtures in plant conservation are Plant Vitrification Solution 2, known as PVS2, and Plant Vitrification Solution 3, also known as PVS3. They are mixtures of various concentrations of dimethyl sulfoxide, also known as DMSO, ethylene glycol, glycerol, and sucrose. DMSO, ethylene glycol, and glycerol are permeating cryoprotectants. Permeating cryoprotectants have two important roles: to stabilize proteins and membranes, and to increase the viscosity of the cytoplasm of the cell. All of these cryoprotectants have small molecules that can easily cross cell membranes. The OH groups on the ends of the ethylene glycol and glycerol molecules are instrumental to the stabilization of the proteins and membranes within the cell. Little is known about the exact role of DMSO. However, it is widely accepted that DMSO helps larger permeating cryoprotectants enter cells. While more research is needed to understand the exact role of permeating cryoprotectants within cells, we can directly visualize cell response using bright field microscopy and the extent of permeation using coherent anti-Stokes Raman scattering microscopy, also known as CARS microscopy. Thus far, we know that cells may respond differently to each cryoprotectant and that each cryoprotectant's distribution amongst and within the cells is non-uniform. Sucrose is also used in cryoprotectant mixtures. It is considered a non-permeating cryoprotectant as its large size prevents the molecules from traveling through the cell membrane. Its role, however, is to surround the cell membrane and balance out the high osmotic pressure gradients created by the permeating cryoprotectants, thereby protecting the integrity of the membrane. Most recent cryopreservation protocols have also included antioxidants. Antioxidants, such as citric acid, reduce the damage throughout various stages of the cryopreservation process. The addition of highly concentrated cryoprotectants causes immense stress on cell membranes, as does the rewarming process. These stressful procedures produce reactive oxidative species that lower the chance of successful cryopreservation. Recent studies have shown that adding antioxidants to any step of the process increases the chance of cell and tissue survival immensely. While PVS2 and PVS3 have been largely successful, exposure times and temperature may need to be optimized when working with different plant species, as each plant may respond differently. Ongoing research will provide more information about the localization, concentration, and functions of cryoprotectant solutions and their components. This will help us to develop shoot tip cryopreservation methods that are robust and effective for long-term preservation efforts.